Okay, it looks like it's about time to get started. Can those joining by Zoom, can you confirm that you have audio and video for me, please? With a thumbs up or just via chat. Looks like nobody's responding. Again, can you just, anybody, can you just continue to pass that around so make sure everybody signs it? Okay, good. Looks like I have audio and video. Okay. So first, a couple announcements before we get to today's lecture. Quiz 10. So I apologize, I forgot about Quiz 10. A couple of you sent me some emails. I didn't realize I hadn't done it until around 3. As you guys probably know, midterm grades were due on Friday at noon. So I got preoccupied with midterm grades, forgot about the quiz until too late. So we'll just put off Quiz 10 till Wednesday and go back to our regular schedule of Wednesday, Friday quizzes this week. And I think that's about all I have. Okay, and I see confirmations that we have audio and video. So let's go back to Chapter 6. Again, like I mentioned on Friday, Chapter 6, I don't think there's any doubt Chapter 6 is the toughest of all the chapters we're going to cover this semester. So again, we're going to take it kind of slow, and I'll repeat myself a lot. So let's just kind of review. So we can split up photosynthesis into 2 sets of reactions. Did everybody find that already? Yeah. Just leave it there then. Okay. What are the 2 sets of reactions that we can divide photosynthesis up into? Anybody on Zoom? Okay, so there's 2 reactions. We have the light-dependent reactions, and then we have the light-independent reactions. Okay. Both of these reactions occur in the chloroplasts. Okay. Again, the chloroplast, remember, is the organelle where photosynthesis takes place. On Friday, we started talking about the light-dependent reactions. Okay, and I realize that these are kind of complex, which is why I said I'm going to take it slow and repeat myself an awful lot. So if we look here, okay, what we're looking at, pardon me, this is the inside of a chloroplast. So remember, the chloroplasts have 3 membranes, right? There's 2 outer membranes and 1 inner membrane called the thylakoid membrane that's folded into stacks of discs. What we're looking at here, this is a cross-section of the thylakoid membrane. Okay, this is the inside of the thylakoid membrane. We call this the thylakoid compartment. Out here, this is the space between the thylakoid and the outer membranes. And you might remember, this is a fluid-filled space called the stroma. All right, again, it's kind of hard for me to draw. Oh, that's horrible. It's kind of hard for me to draw with a mouse. I'm trying my best here. try to do an A again. Okay, so again, this is a fluid-filled area called the stroma, and that A is horrible too. All right, now all of the enzymes and all of the pigments that are involved in photosynthesis are embedded in the inner membrane of the inner membrane of the chloroplast or the thylakoid membrane. So all of these proteins here, these are all proteins that carry out photosynthesis. Okay. Now also recall from Friday that we can split up these proteins into two groups. We have photosystem 2 and photosystem 1. They both have three components in common. So they have a light harvesting complex. So here's the light harvesting complex of photosystem 2. Here's the light harvesting complex of photosystem 1. They have a reaction center that we can't see very well in this particular figure. But the photosystem of photosystem 2 is it's a molecule called P680. It's a modified chlorophyll molecule. And in photosystem 1, the reaction center is a molecule called P700, right? We also have an electron transport chain. So there's two electron transport chains. Here's the electron transport chain of photosystem 1. Here's the electron transport chain of photosystem 2. And what electron transport chains do is they hand off electrons from one component to the next. And again, you can kind of think of these as steps in a set of stairs. So if each component in the electron transport chain is a step, each time the electrons are passed from one step to the next, they go down an energy level. Okay, so the electrons, as they're passed from component to component, they go to lower and lower energy levels, and the cell can capture some of the energy of electrons going to lower energy levels and use that to do work. Okay? In photosystem 2, the work that's done is a proton is pumped from the stroma into the thylakoid compartment. The work that's done with the electron transport chain of photosystem 1 is a molecule of NADP is reduced to NADPH. And remember, NADP is a cofactor. This is one of those molecules that helps enzymes by carrying things such as hydrogens, electrons, and functional groups. Okay, in this case, NADP carries electrons and hydrogens. Okay, so that's a good review. Oh, one more very important component. So that's photosystem 1 over here. So we have the light harvesting complex, electron transport chain, and a reaction center of photosystem 2. So this is photosystem 2 over here. Over here we have the light harvesting complex, reaction center, and electron transport chain of photosystem 1. So remember that photosystem 2 comes before photosystem 1. This is photosystem 1. And over here, this is a molecule called ATP synthase. So ATP synthase, this is actually very important. This is actually going to make ATP. So it's going to take the energy that's been stored up in a concentration gradient while we're doing the light-dependent reactions, and they're going to convert that energy into ATP, which of course is the energy currency of the cell. Whoops. Okay, so let me clean this up and let's talk about exactly what happens in the light-dependent reactions. Okay, so of course the light-dependent reactions are going to start over here with the light harvesting complex of photosystem 2. Does anybody recall what happens to start this whole process? So a light harvesting complex, that's an array of chlorophyll molecules that capture light. So what's the first thing that happens? Anybody on Zoom? Okay, so the first thing that happens is it captures a photon. I see Jared from Zoom. Yeah, so the light harvesting complex, this is just a whole bunch of chlorophyll molecules embedded in the membrane. And it captures a photon of light. Now remember, a photon is a particle of light. Light is one of those weird things in physics. Sometimes it acts like a wave, sometimes it acts like a particle. In this case, it's acting like a particle. So here's the light harvesting complex of photosystem 1. So this is just a whole bunch of different chlorophyll molecules all together. So a photon of light is captured. Anybody recall what's going to happen next after that? Anybody on Zoom? Okay. It reaches the Okay. So Jared again says it reaches the reaction center. Eventually, it's going to reach the reaction center. So this photon is going to bounce randomly between all of these different chlorophyll molecules in the light harvesting complex until it reaches the reaction center, which is a specialized molecule of chlorophyll in the reaction center. Okay. Once it reaches the reaction center, that photon is going to excite 2 electrons in the reaction center, and that's going to eject those 2 electrons into a higher energy state. So when these electrons are in a higher energy state, they're going to be ejected from P680, so they're going to leave the molecule. That means we need replacement electrons. Does anybody, so Jared's answered 2 already, does anybody besides Jared know where those replacement electrons are going to come from? Nobody recalls? H2O, very good. So down here, what P680 is going to do is going to split a molecule of water into 2 electrons. Those 2 electrons are going to replace those 2 that were lost when 2 electrons entered a higher energy state. It's going to break it down into 2 hydrogens and 1/2 of an O2 molecule. Okay, so remember, the thylakoid membrane is a phospholipid bilayer, right? Only small, nonpolar, uncharged molecules like oxygen, O2, can freely cross a phospholipid bilayer. So the 1/2 of an O2 that we get from splitting this can join with another 1/2 of an O2, make an O2, that can diffuse out. Hydrogens, on the other hand, carry a positive charge. Okay? They can't cross the phospholipid bilayer. So they're going to accumulate here inside of the thylakoid compartment, inside the thylakoid discs. Okay, so now let's get back. So we talked about 2 electrons being ejected from P680. They're going to enter an electron transport chain here. Those electrons are going to be passed from one component to the next in the electron transport chain, going to lower and lower energy levels each time. And again, like I said, the cell can capture some of the energy of electrons going to lower energy states to do work in the cell. Specifically, the work that's going to be done by the electron transport chain of photosystem 2 is that a proton is going to be pumped from the stroma into the thylakoid compartment. So it's going to power active transport. Remember, active transport is transport against the concentration gradient, and we need energy in to do it. Okay? That energy is going to come from the electrons passing through the electron transport chain, and the work that's going to be done by the electron transport chain of photosystem 2 is that a proton is going to be pumped from the stroma into the thylakoid compartment. And again, we're building up a concentration gradient here that's going to be very important later, because there's energy being stored in that concentration gradient. Okay, so that finishes up with photosystem 2. We're over here at photosystem 1. Next thing that's going to happen, so the light harvesting complex of photosystem 1, again, is going to capture another photon of light that's going to bounce randomly between the chlorophyll molecules in the light harvesting complex until it reaches the reaction center of photosystem 1, a molecule called P700. Again, that electron, 2 electrons are going to be ejected into a higher energy state. Again, we need replacement electrons, right? Okay, this time, instead of splitting a molecule of water, the electrons from the electron transport chain of photosystem 2 are used to replace the lost ones in photosystem 1. Okay? Again, these electrons, so this time the electrons are going to enter a shorter electron transport chain. So you can see the electron transport chain of photosystem 1 is much shorter than that of photosystem 2. So it's going to enter a shorter electron transport chain. Again, we're going to use the energy of the electrons going to lower energy states to do work. Specifically this time, the work being done is to reduce a molecule of NADP to NADPH. So 2 electrons and a hydrogen are transferred to an NADP, and we get an NADPH. Whoops, that was really bad. Okay, but NADPH, this is going to become important later when we talk about the light-independent reactions. So NADPH is important. Okay, but we haven't made any ATP yet. That comes from over here. Okay, this is a molecule called ATP synthase. ATP synthase, this is a really interesting molecule. If you take cell biology, if you continue with biology as a biology major and take cell biology, your instructor will talk about ATP synthase. It's a really interesting enzyme. ATP synthase contains a channel. So remember that protons can't cross a phospholipid bilayer by direct diffusion, but they can go down a protein channel. Okay, so we have a channel here. Protons are going to go down their concentration gradient through that channel, and as they pass through that channel, some of the energy of them passing through that channel is captured and again used to do work. Specifically, that energy this time is used to drive the reaction of taking ADP plus inorganic phosphate here, ADP plus a phosphate, and making ATP. Okay? So each time a proton goes down that concentration gradient, it can drive the formation of ATP from ADP and inorganic phosphate. And of course, ATP is the energy currency of the cell. So this is very important. And again, just like with any DPH, this is going to come in, this is going to be very important in the light-independent reactions when we're going to make glucose from CO2 and water. Okay, let me just clean this up. We'll continue. Okay, does anybody have any questions on this so far? Okay, let's continue. All right, I didn't quite erase everything. Okay, so this is just another slide, but we're going to go through the same thing. This is another slide showing what happens in the light-dependent reactions. Okay, so again, as you guys know, the first thing that happens to initiate the light-dependent reactions is a photon of light is captured by the light harvesting complex of photosystem 2. Once it's captured, it's going to bounce around there in the light harvesting complex until it encounters what? Anybody know? Anybody on Zoom? So there's 2 of these, 1 for each photosystem. Anybody? Okay. Until it, P680, I see it on Zoom. The reaction center, P680. The Zoomers are stepping up here. Yeah, it's going to encounter the reaction center, P680. And maybe we can get somebody from the class to answer. What's going to happen once that photon encounters P680? Yeah, 2 electrons are going to enter a higher energy state and be ejected from P680. Okay, so here we are. So here's P680 of photosystem 2. So once the photons of light get here, then 2 electrons are going to become excited, and they're going to be ejected from P680. Where are those? What's going to happen to those 2 electrons next? They're going to enter what? The ETC, or the electron transport chain. Exactly. So the 2 electrons are going to enter the electron transport chain, and those electrons are going to go to lower and lower energy states. Now on this slide, it doesn't show what happens. Remember, the cell can capture some of that energy and use it to do work. And on this slide, it doesn't show exactly what work is being done by these electrons. What's the cell going to use that energy for? Anybody on Zoom? So they can capture some of this energy of electrons going to lower energy states. And I don't see anybody on Zoom. Okay. This is going to be used to power a proton pump. Okay, and that proton pump is going to pump a proton from the stroma into the thylakoid compartment. So again, we're building up a concentration gradient of protons that we can use at the end to generate ATP using ATP synthase. Okay. There's one thing I skipped over. So we have these 2 electrons in a higher energy state. We need to replace those 2 electrons in P680. So where do we get those 2 replacement electrons from? We split a molecule of water into 2 protons. Remember, those 2 protons are going to be stuck there in the thylakoid compartment because they can't cross the phospholipid bilayer. 2 electrons are going to replace the 2 electrons that are lost. 1/2 of an O2. That'll join with another half an O2. That can diffuse out because it's small, nonpolar, uncharged. Okay. All right. So now we're over here at photosystem 1. Again, the light harvesting complex of photosystem 1 is going to capture a photon of light. That photon of light is going to bounce around randomly until it encounters the reaction center of photosystem 1, which is P700. And then again, what happens? So 2 more electrons. Yeah, 2 more electrons are going to be ejected into a higher energy state. Okay, 2 electrons are ejected into a higher energy state. Again, they're going to enter an electron transport chain. This time, it's a shorter electron transport chain. Can anybody tell me what happens at the end of this electron transport chain? What work is going to be done? So we're going to reduce what? Okay, so at the end of this shorter electron transport chain, a molecule of NADP is going to be reduced to NADPH. Okay. Then, of course, after that, as the light-dependent reactions proceed, we build up this concentration gradient, this high concentration of protons in the thylakoid compartment. ATP synthase can capture some of the energy of these protons rather, passing through the channel in ATP synthase and use that energy to make ATP from ADP and inorganic phosphate. The ATP, again, it's adenosine triphosphate. So if we take ADP, adenosine diphosphate, add a phosphate, we get ATP, adenosine triphosphate. Okay, so hopefully, like I said, I was going to repeat myself. I think that's about, I went through it 2 times today and probably 2 times on Friday. Is everybody starting to get how the light-dependent reactions work? I know it's complicated. You might want to go over the recorded lectures a couple times to really get it to sink into your head. Let me ask, are there any questions on the light-dependent reactions before we start talking about the light-independent reactions? Okay, I'm not seeing any questions from Zoom. So let's continue then with the light-independent reactions. So again, these used to be referred to as the dark reactions. The reason we don't call them the dark reactions anymore is because although they can happen in the dark, it doesn't have to be dark for these reactions to happen. They actually happen all the time, regardless of whether there's light available or not. So we refer to them now as the as light-independent reactions. Another name for these reactions is also called the Calvin-Benson cycle. That's named after the scientists that discovered it, Calvin and Benson. And there's a new term here we should probably talk about. So this is called the Calvin-Benson cycle. A cycle is a specific kind of metabolic reaction. Okay, I'm going to go ahead and write on the board because I'm not very good at writing with the mouse on Zoom. Okay, so the light-independent reactions are sometimes called the Calvin-Benson cycle. So they're a type of metabolic reaction known as a cycle. And what differentiates a cycle from other metabolic pathways is that you start with the same molecule that you end with. So at the end of the cycle, the molecule that you started with is regenerated, and then it goes back into the cycle again. So a cycle this is a type of metabolic pathway. That ends with one of the same molecules that you begin with. Okay, so in a cycle, we end with one of the molecules that we begin with. So we'll see this again. We'll see this in aerobic respiration as well. There is part of aerobic respiration. There's some reactions called the Krebs cycle. And again, with the Krebs cycle, you regenerate one of the molecules that you start with, and it goes back into the cycle. Same thing with the light independent reactions or the Calvin-Benson cycle. At the end of this metabolic reaction, you regenerate one of the molecules you started with, and then it just goes back and keeps going round and round and round, hence the term cycle. Can you move the camera so that we can see on the board? Yes, I'm sorry. When I have the, let me just stop the share for a second. When I'm sharing my screen, I can't see myself. So if you can't see something, let me know. Oops. Oh boy. Okay. There, hopefully you guys can see that now. Hopefully you can see it better than me, because on my screen that's actually written backwards. Oh no, I can see over here it's actually written correctly. Okay, so a cycle is a type of metabolic pathway that ends with one of the same molecules that it began with. Okay, I'll leave that up for a bit, and then I'll fix the camera later. Let's go back to the PowerPoint. Okay, so the dark reactions of the Calvin-Benson cycle. This uses some of the things that we made in the light-dependent reaction. Sorry, guys. Let me leave that up in case anybody on Zoom is still copying it. This uses NADPH and ATP from the light-dependent reaction. So like I said, we're going to use the things we made in the light-dependent reactions in the light-independent reactions. This also requires CO2. Okay, CO2 is very important. What we're going to do in the light-independent reactions is we're going to make glucose. So remember, photosynthesis is a biosynthetic reaction. So we take smaller, lower energy molecules and use them to make bigger, higher energy molecules. So in this case, we're going to take small, low energy molecules like CO2 and water. Okay, specifically, CO2 is going to be the source of carbon for us, and we're going to make bigger, higher energy molecules, in this case, glucose. Okay, glucose is, the chemical formula for glucose is C6H12O6. So this is a bigger, higher energy molecule. Okay, so I'm going to assume everybody zooming in has copied down the notes, and I'm going to put the screen back down. Okay, so in the light-independent reactions, ATP provides energy for the reaction. So remember, this is biosynthetic, this is endergonic, so we need to put energy in. We need something to drive these endergonic reactions forward. In this case, it's going to be ATP from the light-dependent reactions. NADPH acts as a donor of electrons and protons. Okay, so the electrons and protons from NADPH are going to help us to make glucose from CO2 and water. CO2 is a source of carbon and oxygen. And this is something that always kind of blew my mind. So the carbon that goes into glucose comes from the air. So just remember that the next time you're putting sugar in your coffee or having spaghetti, is that the glucose that makes that up, at one time the carbon that went into that glucose was in the air, which I think is, for some reason, that always kind of amazes me. All right, so we're going to use energy from ATP to provide energy from the light-dependent reactions. We're going to use NADPH from the light-dependent reactions to provide electrons and protons. We're going to use CO2 from the air as a source of carbon and oxygen to make glucose. The end products of the light-independent reactions are an H2O, and a phosphorylated glucose. This simply means a glucose that has a phosphate attached. Now, the plant cell can then take that phosphorylated glucose and do a number of things with it. It could use it to make bigger, more complex carbohydrates. So it can use it to make starch. It can use it to make disaccharides. Okay, or it could use it directly as energy. Remember, glucose is a primary energy source. Okay, so the end products are a phosphorylated glucose and water. Okay, so here's the phosphorylated glucose right down here. Okay. So before we go over through the step chart, let me explain exactly what happens here. What we're starting out, actually I'm going to write this down on the board. So as you can see from this figure, there's 3 steps to the light independent reactions or the Kelvin-Benson cycle. We have, I'm not sure what I did there. Okay, so we have the carbon fixation step. That's where we take carbon dioxide from the air and we join it to another molecule so it can enter the cycle. We have the reduction. What was that? We have the reduction of 3-phosphoglycerate, glyceraldehyde 3-phosphate. Does somebody have a question? Somebody might have their microphone on. Okay. Then the third step, we have the regeneration of ribulose bisphosphate from glyceraldehyde 3-phosphate. Okay, this is where we regenerate the molecule that we started with so it can go back into the cycle. Okay, so let's look at each of these steps individually. Okay, so we have the light independent reactions. The first thing that happens is we're going to start with a 5-carbon molecule of ribulose bisphosphate and a 1-carbon molecule of CO2. Okay, ribulose bisphosphate, that's over here. Whoops, I didn't mean to do that. Okay. So RUBP, this stands for, you don't have to know this, if you just know RUBP, that's fine. RUBP stands for ribulose bisphosphate. Okay. This is one of the molecules that we start with in the light independent reactions, and this is also the molecule that we regenerate at the end so it can go back into the cycle. Okay, and you can see each of these red dots represents 1 carbon. So 1, 2, 3, 4, 5. So we're starting with a 5-carbon molecule of ribulose bisphosphate, and we're going to join that to a 1-carbon molecule of CO2. So we're going to start with a 5-carbon ribulose bisphosphate, or RUBP, and a 1-carbon CO2. So when we join the 5-carbon ribulose bisphosphate together with the 1-carbon CO2, we get a 6-carbon molecule, obviously. However, that 6-carbon molecule is very unstable, and it almost immediately breaks into 2 3-carbon phosphoglycerates. So we get a 6-carbon molecule that breaks into 2 3-carbon phosphoglycerates. Okay, and phosphoglycerates, often this is just abbreviated PGA. If all you know is, if all that you remember is PGA, if you don't remember phosphoglycerate, that's just fine. Okay, so we're right here. We're in the first step, fixation of carbon dioxide. So we join a 1-carbon CO2 with a 5-carbon RUBP. we get an unstable 6-carbon molecule that breaks into 2-PGA, or 3-phosphoglycerate. That brings us to this next stage, reduction of 3-phosphoglycerate to glyceraldehyde 3-phosphate. So in this step, next, we're going to use the energy from ATP and we're going to use the electrons and hydrogens from the NADPH that we made in the light-dependent reactions. It's a little messy. So energy from ATP, electrons and hydrogen from NADPH. To reduce phosphoglycerate to phosphoglyceraldehyde. Phosphoglyceraldehyde, we abbreviate this PGAL. If you guys just remember PGAL, you don't have to remember the whole big word. But PGAL. Okay, so that brings us down here to the bottom of the cycle. Now, there's two things we can do with PGAL. We can take 2 3-carbon PGALs, join them together, and use them to make a glucose. Okay? However, we still need to regenerate that 5-carbon ribulose bisphosphate so it can re-enter the cycle. So we're not going to use all of the PGAL to make glucose. We're going to use some PGAL to make a phosphorylated glucose. Okay. Then we're going to take some of this PGAL, and we're going to regenerate the ribulose bisphosphate from G3P is just another word for it. I prefer the abbreviation PGAL. G3P is just another abbreviation for PGAL. Okay, we're going to take some of that PGAL, use the energy from ATP, and regenerate the 5-carbon ribulose bisphosphate. Okay, let me adjust the board for the people on Zoom. All right, so let's zoom out. Okay, folks, I'm going to kind of continue over on the left-hand side of where I was writing. Okay, so number 4. You guys see that? I have a little more room actually. Number 4. Some PGAL is used to make phosphorylated glucose. And number 5, some of the PGL plus ATP from the light-dependent reactions is used to regenerate the ribulose bisphosphate. Okay. So if you remember these 5 steps for the light independent reactions, you're good for the exam. Okay, so we start with a 5-carbon ribose phosphate, a 1-carbon CO2. We make a 6-carbon molecule that breaks into 2 PGAs. We're going to use the energy from ATP and electrons and hydrogens from NADPH to reduce that PGA to PGAL. We can use PGAL to make glucose directly, but we got to save some of that PGAL and use it to regenerate the 5-carbon ribose bisphosphate. Okay, and that's going to take more energy from ATP. All right, and I see that we are out of time. So again, remember, we're back on our regular schedule of Wednesday, Friday quizzes this week. Sorry about quiz 10 on Friday. And I think that's about it. So I will see you guys either on Zoom or in class on Wednesday. Okay, Zoomers, do you need more time to copy notes, or can I end the... Just give me like a thumbs up or a chat if you need more time for taking notes. All right, if you don't need any more time for taking notes, I am going to end the meeting, and I'll see you guys on Zoom or in class later this week. So long.